This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is polymerase chain reaction? Answer: polymerase chain reaction is a molecular biology lab procedure used to amplify a desired fragment of DNA. Question: what is the use of polymerase chain reaction? Answer: polymerase chain reaction is useful as a diagnostic tool, such as for neonatal HIV and herpes encephalitis. Question: how does polymerase chain reaction work? Answer: the polymerase chain reaction amplifies a specific DNA fragment using a heat-stable DNA polymerase, specific primers, and repeated cycles of heating and cooling. Question: what are the steps involved in polymerase chain reaction? Answer: the steps involved in PCR are denaturation, annealing, and extension. Denaturation step involves heating the DNA to separate the two strands. Annealing step involves cooling the DNA and adding primers that bind to specific sequences on the template DNA. Extension step involves heating the DNA again to allow the polymerase to extend the primers, creating new DNA strands. This process is repeated multiple cycles to achieve the desired amount of amplification. Question: how is polymerase chain reaction used in diagnostic procedures? Answer: PCR is used in diagnostic procedures by amplifying a specific fragment of DNA associated with a disease or condition. The amplified DNA can then be detected using various methods such as gel electrophoresis, fluorescent probes, or by sequencing. Question: what are the advantages of using polymerase chain reaction? Answer: the advantages of using PCR are high sensitivity and specificity, ability to amplify small amounts of DNA, and the ability to detect rare genetic variants. Question: are there any limitations of using polymerase chain reaction? Answer: yes, there are some limitations of using PCR. One limitation is that PCR can only amplify specific sequences, so it may not detect all forms of a particular genetic variant. Additionally, PCR can produce false positives or negatives if proper controls are not used or if the DNA sample is contaminated. Question: what are some applications of polymerase chain reaction? Answer: some applications of PCR include forensic science, genetic engineering, medical diagnostics, and research in fields such as evolutionary biology, ecology, and epidemiology. Question: are there any alternatives to polymerase chain reaction? Answer: yes, there are alternatives to PCR such as ligase chain reaction, transcription-mediated amplification, and nucleic acid sequence-based amplification. These techniques share similar principles with PCR, but they have different specificity and amplification efficiencies. Question: how has polymerase chain reaction evolved over time? Answer: PCR has evolved over time to make the process faster, more efficient, and more specific. Advancements include the development of thermostable polymerases that can withstand high temperatures, new primer design strategies, and the use of fluorescent probes for detection.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.